talk. It is my honor to introduce to you Professor Paola Picotti. She is professor at the ETH in Zurich for molecular system biology. After completing her PhD at the University of Padua in Italy, she moved to Zurich to do her postdoctoral research. And then in 2011, she was appointed assistant professor, still at the ETH. And then in 2017, she was promoted to associate professor. Professor Picotti's current research focuses on how the conformational changes of protein impact the cellular networks. She is using her innovative proteomic techniques to unveil the dynamic 3D proteomes in complex environments, with the goal of understanding the mechanism underlying protein aggregation in neurodegenerative diseases. Unfortunately, Professor Picotti cannot be here today, but she will join us through Zoom connection.
Thank you very much, Carlo. Thank you also, Jürgen and colleagues, for inviting me to give this presentation. I also want to thank you for giving me the opportunity of presenting remotely. I apologize for that. It would be so much better to be there with you, to have in-person interactions, and also to enjoy the city of Barcelona, which I love very much. But here it's a really packed week, and there are a number of institutional commitments for me in Zurich, so unfortunately I cannot be there. Okay, so as you heard, my group works in the field of proteomics since a few years, and recently we also took a turn towards structural proteomics. And with my presentation today, I would like to propose that structural proteomics tools are now mature enough to branch out of the world of structural biology and can be used as new tools in functional proteomics screens. So the screens many of us are interested in. And I will also propose that this new tool will increase the coverage in detecting alterations, any kind of alteration, physiological alterations or pathological alterations, and it will also help us generate mechanistic hypotheses out of our large-scale proteomics data. This is the thesis for today, and now I will go step by step. And I will present a mixture of published, but also quite a bit of unpublished data. So as many of you know, and as you most likely heard during this course, proteomics has been used a lot in the literature to study what I call here complex phenotypes. For example, how cells respond to a perturbation or how human disease develops. And at the very beginning, when we know nothing or very little about the responses to a perturbation we are interested in, it's typically a good idea to ask the question, which nodes in a complex physiological network are involved in the response to the perturbation we are interested in. And when the nodes in this network are proteins, then one way to do that is to conduct one of our favorite proteomic screens. And these yield typically lists of proteins that change in abundance in condition B relative to condition A, together with the associated fold changes. And I see, and I am sure you discussed this a lot in the course. And then one thing we do often is to conduct some kind of clustering analysis to identify groups of proteins that behave the same, minimally proteins that go up and proteins that go down. And then we can also do some kind of functional enrichment to ask if there's anything in common between proteins in the same group. For example, if they are associated to the same biological process or they are part of the same pathway. And if we find something enriched here, then we speculate that these processes or pathways are involved in the response to the perturbation we are interested in. Now, if you look at this slide, exactly this approach has been used in hundreds of studies in the literature, in many also supported by Max Quandt, which has been really instrumental in making this analysis rigorous, fast, and also user-friendly. And we have used it also a lot in our group. However, if you have done yourself one of these analyses, or if you will do one of these analyses, there are a few issues. And one issue we certainly bumped into very often is the first one described on this slide. Once you have your list of regulated proteins, it's typically very difficult to automatically generate a molecular hypothesis on what could be going on in the cell. So it's difficult to develop experiments to follow up a specific speculation you generate just out of a list of regulated proteins. The second issue is that the abundances of proteins don't necessarily change when pathways are regulated in a cell, because pathways and protein function in general can be regulated by a number of other events. And an example I like very much and I show often is the one you see here. This is a pathway regulated in yeast cells under conditions of osmotic stress. It starts up here when cells sense the increased salt concentration in the medium. They activate a cascade of phosphorylation events. Then a protein goes into the nucleus and it gets engaged in protein-protein interactions. Also protein-DNA interactions are triggered. Then these two proteins become phosphorylated downstream of this cascade. This enzyme produces a metabolite, which then in turn binds allosterically to this other protein here. So you have also a metabolite binding event. And the result of all these changes is that the levels of substrates in this pathway you see here change, especially they decrease in this branch here, and they increase down here. So that basically fluxes are shifted away from the production of pyruvate into the production of glycerol, which is then used by yeast to counteract the stress. Now, forget about the details, but look at this picture one moment. None of these changes would be captured easily by a classical proteomic analysis where you measure protein abundances. And in fact, protein abundances before and after osmotic stress on a short time scale, let's say 5 or 10 minutes, abundances stay pretty much the same. So we started asking the question, is there a way to capture simultaneously all the changes you see on this slide? You see they are very different molecular events, post-translational modifications, binding events, and so on. Is there a way to capture them all within the same analysis? Do they have anything in common? So if you think about it, there is something they have in common. Binding events, PTMs, protein-DNA interactions, aggregation, these are other things I did not mention. They all have something in common. They all locally or globally affect the structure of the protein involved. So we thought at one point that by detecting altered structural states of proteins, we may be able to detect altered functional states on a proteome-wide scale. So this is basically the old classical paradigm. Structure of a protein determines the function of a protein. But here the idea is to exploit this principle on a large scale to detect functional alterations in an unbiased manner. So that's the concept. So how can we detect structural alterations of proteins? We developed a few years ago now a method we call the limited proteolysis coupled to mass spectrometry. Some of you may be familiar with it. So it starts with a native protein extraction. So basically we lyse cells or tissues in a way that preserves protein structures. And then we add for a very short time a non-specific protease. We like very much proteinase K for this step. And then we very quickly quench the reaction. So the idea is to stop the reaction after the very first cleavage events. So we don't let the digestion go to completion as you do in a classical trypsin digestion experiment. Now, these fragments are structure specific because under these conditions, the protease will cleave at the most flexible and accessible regions in a protein. So if the structure of the protein changes, you will see different fragments here, but they are still a bit too large for bottom-up mass spectrometry, so peptide-based mass spectrometry, again, the type of mass spectrometry you use in shotgun proteomics or DIA proteomics. And so what we do is to then denature these fragments, and then we digest them again with trypsin to generate peptides. So basically, we produce shorter fragments that still retain the structural information. And then we measure these peptides by mass spectrometry. And here I have to say different approaches work. You can do DDA, DIA, SRM, so they all work depending on the question. So bottom line is this technique generates proteolytic fingerprints that change when the structure of a protein changes. And we generate these fingerprints for any protein in a lysate that we can reasonably detect by mass spectrometry. Then we translate the fingerprints into what we call now structural barcodes, which is basically the intensities of the fragments mapped along the sequence of a protein from the N-terminus to the C-terminus. And then when we apply a perturbation to our cells, we compare the barcodes before and after, and this tells which protein underwent a structural change and also where exactly along the sequence the change occurred. So when we started working with this technique, it was year 2013 or so, we were very much interested in protein aggregation. That's the second research line of my lab. And so we very much wanted to know if we could use this technique to detect the formation of protein aggregates in cells and tissues. And so we focused on the protein we were working on. This is called alpha-synuclein. I'm sure many of you know it. It's a protein involved in Parkinson's disease, and it's thought to form amyloid aggregates in the brain of Parkinson's disease patients. But in fact, the story is much more complex, and now I will show you some recent data on this story. So in fact, alpha-synuclein exists not only as an unfolded protein, so that's what happens if you solubilize the protein in a neutral buffer. It's an intrinsically disordered protein. But then if you add lipids, it becomes alpha-helical. If you wait, if you give your sample enough time, the monomer will aggregate into amyloid fibrils spontaneously, like in about a week. And on path to the formation of amyloid fibrils, it can even acquire different types of oligomeric states. So it's a very complex protein. It has also been called a chameleon protein due to this heterogeneity. Now, all the structures you see here on the left are the structures we know from in vitro experiments, starting from the purified protein. But nobody really knows which of these structures is present in a cell. There are various hypotheses in the literature, but it's still quite controversial. So we asked, can we learn which of these structures is present in cells and tissues using our approach? So first, we generated the structural barcodes for each of these structural states. You see them here. It's about 250 peptides along the sequence of the protein. And then we asked whether the barcodes you see here are sufficiently different. So whether they distinguish the 5 different states of alpha-synuclein. Here you see a principal component analysis. Same color means same structure, and the different points are replicates of the same prep. So we started with different preps. And on the right part of the slide, you see the peptides we measure along the sequence of alpha-synuclein. You see there's a very high degree of redundancy. For a given region, we cover typically many peptides mapping to the same residues. So the main message from this PCA analysis was that yes, we can differentiate quite well the different structures, but the main question remained, which of these is present in a cell? So we thought of using the following approach. We have the fingerprints or the barcodes from in vitro protein structures, but we can in principle acquire exactly the same fingerprints from a real sample, from cells or from tissues of patients, for example. And then what we can do is simply to compare in vitro and in vivo or in situ fingerprints and ask which of these structures we know from in vitro studies is present in cells and tissues. In fact, again, this is not as simple. We applied a linear combination approach, which means we asked which linear combination of fingerprints from the in vitro structures results in the fingerprint we detect in cells and tissues. So basically, we allow for the coexistence of multiple structures, possibly in equilibrium with each other. So first, we asked whether this whole idea of linear combination worked at all. And so we worked with a very controlled sample. We prepared mixes of 2 in vitro structures, for example, 50% monomer, 50% oligomer, or 50% monomer, 50% fibril, and so on. And then we did LIP, MS, on each of the mixes. And then we applied the linear combination approach. So we asked which linear combination of proteolytic fingerprints from the pure structures results in the fingerprint we detect for the mixture. And here you see the result of the prediction. So if you look at this slide for a few seconds, you probably realize that in all cases, the approach captured the two correct components. For example, here the prediction is around 50% monomer, 50% oligomer, and that's exactly the composition of the mixture. But we still struggle a bit with quantitative accuracy. For example, look down here. Here the mixture was 80-20, and the prediction is 70-30. So there is still a quantitative error, but the main components are correctly captured. So based on this, we learned that you can detect different conformational states of a protein, and if template structures are available, so structures that have been well characterized in vitro, you can use this approach to deconvolve the structural heterogeneity of a protein in a mixture. Now this is cool, but again, it's still in vitro. What about the real complex samples? So we first looked at yeast cells overexpressing alpha-synuclein. They produce these inclusions, and these have been thought for long to be aggregates, and then this hypothesis has not been confirmed. And so the question remains, what are these blobs? And so we applied the same approach. We acquired the fingerprints, and then we applied the linear combination approach. And the prediction is that alpha-synuclein in these cells is mostly alpha-helical, to a certain extent also in a monomeric unfolded state, but we also detect the presence of amyloid fibers. Now, it's very interesting that we detect the helical structure of synuclein here, the one you obtain when synuclein binds to lipids, because if you stain these cells with dyes for lipid droplets, these are lipid deposits in the cell, the staining is really overlapping perfectly with the alpha-synuclein stain, suggesting that these blobs are not aggregates of alpha-synuclein, they are lipid droplets, most likely coated with alpha-synuclein that acquire the helical structure upon binding to lipid droplets. So that's a prediction, and what I can say for the moment is that this is in line with the biochemical analysis we did in parallel on the same cells, for example, size exclusion, fractionation of the lysates, filtration, and native gels, which predicted pretty much the same distribution of molecular weights. And then we moved on to analyzing brains from healthy individuals and Parkinson's patients. And there, the helical structure of synuclein is detected even more predominantly, as you see here from the orange bars, suggesting that these might indeed be the physiological structure. And this is also in line with the recent mutagenesis scanning experiment, which identified mutations that affect toxicity of alpha-synuclein in yeast cells. And these mutations have a very repetitive pattern compatible with the presence of a helical structure. So that's where we are at the moment. And the main take-home message from a technological or methodological point of view is that by mass spectrometry, we can quantitatively deconvolve a range of very different structures in situ. So this range from an unfolded structure all the way to very compact amyloid fibers. And this is something you cannot do with high resolution techniques. So that's where a niche for mass spectrometry is, to do these types of analysis. Okay, but in this analysis, I have to say we don't see only synuclein because we do a global screen. So we acquire fingerprints for all other human proteins we detect in the same samples. And so we exploited this capability of the method to analyze the inter-individual variability of protein structures. So how much the structure of a given protein varies across different humans. And for that, we used samples from a healthy cohort, and we use, in this case, biofluids. And it's very interesting. I just tell you the main message without going into the details. Some structures are incredibly stable across individuals. The fingerprints are identical. Some structures vary only at one specific spot, for example, binding site, and others are completely different across individuals. So very global variability across different individuals. Now, back to the initial questions, we asked whether this technique could detect amyloid formation, and that's the case, and we learned that it can also detect different conformational states of a protein. So next we asked, can it detect also a very subtle structural alteration? For example, an alteration due to the binding of a small molecule, which should, in principle, only cause a very small alteration at the binding site, minimally by shielding the binding site. So to address that, a few years ago, we performed experiments where we lysed the cells, and then we added a specific compound to the lysate for which we knew the binding partners, the target, the binding protein. And then we measured the barcodes with and without the compound, and we asked those that change the barcodes. Are those proteins that bind that are known to bind the compound? And again, I will show you now some old data before I move on to the new data. This is an old experiment we did with the drug cerulenin. This is an antifungal compound that binds the protein fatty acid synthase 2, subunit 2. So here we lysed yeast, we added cerulenin, and then we measured our fingerprints with and without the drug for roughly 2,500 proteins. And of these, only the fingerprint of cerulenin changed, of fatty acid synthase changed. For this protein, we detected more than 50 peptides, and only one peptide changed dramatically, and that's the peptide you see here in red. So we mapped the peptide to the structure of the protein, and we noticed that it was the peptide at the binding site of the drug, suggesting that you can use this technique to detect the targets of a compound, but also the binding site. So this was really one of the very first experiments, but now I have to say, after some years of applying this technique, not all experiments are so clean. This was a very clean experiment. Sometimes you don't see a single peptide. Sometimes you see a cluster of peptides around the binding site. Sometimes you also see peptides at completely unrelated regions. And these cases are most likely conformational changes induced by binding of a molecule. So in those cases, when you see peptides all over the place, there's no way to pinpoint the binding site of the compound. But in many cases, it is possible when you have only a few localized changes. So when I started to show this data, then I started to receive two questions. Okay, it works for small molecules. Does it work also for proteins? So can you add a protein to an extract and identify binding partners using this approach? And the second question we received a lot was, does it work also for membrane proteins? Because these are very tricky, they are very hydrophobic, they are often lost in our experiments, so does it work there? So we gave it a try, and we focused on the known interaction between the big transmembrane protein adenylyl cyclase 8 and the protein calmodulin. Calmodulin is known to bind AC8 at the two blue binding sites you see here. And so here we worked with a crude membrane prep. Basically, we lysed the cells, we spun down membranes, we dumped the supernatants, and then we resolubilized the membranes, and we added calmodulin. And then we compared again fingerprints of all the proteins we detected plus minus calmodulin added to the membrane prep. Now you see down here the coverage for adenylyl cyclase. Every blue bar is a peptide. As you can see, the coverage is pretty good, but we clearly do not detect at all the transmembrane regions. That's a typical problem. These peptides can be really hydrophobic. They can get lost in the pipeline. But the rest of the sequence is relatively well covered. And then when we added calmodulin, 3 peptides of all the blue ones you see here changed, and changed proportionally to the amount of calmodulin we added. And these peptides were located at the known binding sites of adenylyl cyclase 8. So far, so good. So we thought even for protein-protein interactions, this can work. Now we have done a few of these analyses, and my impression is that it could work a bit better for protein-protein interactions than for protein-metabolite interactions, because the binding site is often a bit bigger when two proteins interact. But that's just our initial gut feeling with these experiments. And then next we asked, good, if it works for protein-protein interactions, then maybe we can use it for mapping epitopes of antibodies. So basically to tell which regions of a protein bind an antibody for antibodies for which such epitopes are not known. And so we tested this idea with a set of antibodies that have been recently developed by the group of Bruno Correa and others. I think one antibody is not from Bruno, against the protein of the respiratory syncytial virus, the fusion glycoprotein. So this protein exists in two conformations. You see them here. One occurs before fusion of the virus with a mammalian cell membrane. The second one occurs post-fusion. And the three antibodies are available. The blue one only hits the pre-fusion glycoprotein. It does not bind the post-fusion conformation. The red and the yellow one bind both conformations, but at two completely different epitopes. So the epitopes are known for these antibodies. And so we focused on the post-fusion glycoprotein. And then we added the three antibodies individually and performed the LIP. So now you see the results of this analysis. When we added the blue antibody to the post-fusion protein, so this is the protein it does not bind, we did not see any altered. When we added the red antibody, which is supposed to bind the protein, we detected, a few green peptides. And these peptides were located inside the known epitope. When we added the yellow antibody, which is also known to bind this protein conformation, we detected a few proteins within the epitope, but also a few, peptides, but also a few peptides outside the epitope. So what we think here is that these antibodies are big, they're really big. And so when an antibody binds here, probably it creates some steric hindrance also at this region. And so this region is less accessible to proteolysis by proteinase K. So as you can see, you hit the epitope, but here the resolution is a little lower than in the red case. But still, overall, it can give an indication on where the epitope is of an antibody. We repeated these experiments with purified proteins, but also we spiked these proteins into a complex lysate of HEK cells. And there we also asked, are these antibodies specific, as the group who developed these antibodies thought, and luckily they were. So this antibody, when we added it to the extract of human cells, we detected 4,300 proteins, and only 6 peptides from these proteins changed, 5 of which luckily were from the expected target of the antibodies. So one could use this technique to assess the specificity of an antibody to be used for purposes and to identify epitopes. So to summarize, first summary, you can use this technique to pinpoint conformational changes of proteins, binding events involving proteins and other proteins, or proteins and small molecules. You can detect aggregation events. I did not show it, but you can also use it to probe unfolding. For example, we increased the temperature of a lysate, and then we looked at the intensities of lip peptides for different proteins, and basically the intensities of these peptides track the unfolding process of these proteins. Good, so now back to our initial picture. We were wondering, can a structural readout capture all the different types of molecular events that regulate protein function? Well, based on these experiments I've shown you until now, it seems that it can capture some of them individually, like this one, protein small molecule binding events, protein-protein interactions, alternative conformations, aggregation. But the question we had at the beginning was a bit different. The question was, can it capture all of them simultaneously within the same analysis? So we addressed this question, going back to the pathway I've shown you at the very beginning. This is the osmotic stress pathway in yeast, because this is concomitantly regulated by many of these molecular events. And so we asked, can we capture them all within the same analysis? We grew yeast cells under conditions of osmotic stress to activate the pathway you have seen. We also grew them in parallel under conditions of heat shock to assess the specificity of the readout. And then we measured in parallel protein abundances. So this was done with the classical shotgun proteomics approach. And in parallel, we measured structural changes of proteins under these conditions. Now here you see a summary. After 10 minutes of perturbation, we detect only 30 or 50 proteins that change in abundance. And this is normal because the time scale of the perturbation is quite short in the minute range. So we don't expect the cell to be able to regulate much protein abundance. But protein structures change much more. As you can see, 300 proteins here, 600 proteins here. So apparently, these other types of molecular events that are captured by structural changes occur on much faster timescales. And then we did one of these geo-enrichment analysis. When we looked at protein abundance data, we didn't see a lot of terms enriched. So you see here on the right side of the plot, geoterms, and then the rectangles show enriched terms, and the Q value is represented by this color scale. So abundance changes did not capture a lot of enriched terms, and that's again normal because the number of regulated proteins is low. But when we looked at protein structural data, many terms appeared enriched, and they were somehow specific because some appeared only for heat shock and some only for osmotic stress. And many of these boiled down to the known suspects, for example, protein folding for heat shock, or the glycerol biosynthesis pathway for osmotic stress. So this pathway is exactly the pathway we wanted to stimulate. And so we mapped the structural data to this pathway. And you see now in orange all the proteins that change structure under these conditions. The two in gray we did not capture, so we detected only a couple of peptides for these proteins. And then we mapped the data to protein sequences. And we noticed that the structural alterations were very much localized to only one spot or a couple of spots in a protein sequence. So initially here we were a bit puzzled because we expected much larger changes. But then we mapped the data to protein structures and things became a bit clearer because we realized that many of these changes were located at functional sites of a protein. For example, for proteins that undergo protein phosphorylation, we often captured peptides containing the phosphorylation site. If you're interested in this aspect, I can give you more information on the statistics of this global analysis. For the protein that was binding a metabolite, we detected the peptide at the metabolite binding site. And down here, maybe you remember I told you these proteins, these are enzymes for which substrate levels change a lot before and after osmotic stress. And here we detected one or a couple of peptides at active sites. So we didn't know exactly what to expect here. So we thought maybe we see active sites because we probe the occupancy of the active site, which means how much of the substrate is bound to the active site after lysis, because remember, we have unfortunately to lyse cells here. And so we performed experiments in which we added the substrates, not only of these enzymes, but of also other enzymes to lysates. And then we monitored the intensities of the orange lip peptides, so the peptides at active sites. And the curves we obtained were very much reminiscent of binding curves, suggesting that most likely this peptide report indeed on the occupancy of the corresponding binding site. So to summarize, we believe, based on these experiments, that this approach captures different types of molecular events with the resolution of single functional sites. And to make another summary here, based on that, we believe that protein structures integrate various types of molecular events that affect protein function. That's why detecting altered structures allows detecting altered functional states on a large scale. And now we recommend to always perform in parallel a quick structural proteomic analysis when you're interested in probing functional alterations beside measuring protein abundances, because you drastically increase the coverage in detecting alterations. I also indirectly proposed a new workflow to generate hypotheses. This workflow starts up here. You do a global structural analysis, for example, based on LIP. You generate dynamic data, so structures that change, and then you map this data onto high-resolution static structures. These are the structures you find in the PDB database that have been resolved, or predicted structures, for example, by tools like AlphaFold. Wherever this is available, you can incorporate biochemical knowledge, but this is not compulsory. And this triad helps to identify which functional sites on a protein are affected by the perturbation you were interested in. So not only which proteins, but you increase the resolution of your analysis, so you can learn whether the site of binding of a metabolite is altered, or the site of a phosphorylation site is altered, whether the active site of an enzyme is altered. And this helps generate testable biochemical hypotheses, as I've shown you for the substrates of the osmotic stress pathway. So this can be very useful to then fine-tune the design of follow-up experiments. It's also useful because if you know exactly which site is altered, you can try and introduce mutations right there to see if this affects the phenotype you're interested in. Now, we are using this pipeline, exactly the pipeline you see here, in a variety of projects. And now I will spend the last few minutes telling you where we are going next. So we realized in all these experiments that we always struggle with one aspect. We can never really disentangle protein-protein interactions from LIP data. So when we do one of these screens, we cannot tell which of the alterations we detect are due to altered protein-protein interactions. And so we asked, is there a way to tell? So we thought, to tell if a LIP change occurs at an interface of a protein complex, we should know where the interfaces are. And this is known only for a few cases, those for which the structures are available. And so we came up with the following idea. We thought if we can somehow separate the completely ensembled state of a protein complex from a partly ensembled state, from even the monomeric subunits, and then if we do LIP on each of these structures, in principle, we should only see differences at the interfaces, because these are the only sites that change in terms of accessibility. And this principle has actually been demonstrated in the past for single protein studies. So we asked, is there a way to separate these three states of proteins? And so we came up with the following pipeline. We take a cell lysate and then we load it on top of a filter with a high molecular weight cutoff. We then collect the L weight and we load it onto another filter with a lower molecular weight cutoff, and so on. So this is a serial ultrafiltration approach. We collect the different fractions and then we do limited proteolysis on each fraction. And the idea is, if a protein is present in multiple fractions, it's probably present under different assembly states. And so when we do LIP and we compare the digestion patterns for the same protein, for example, the red protein here across samples, then the differences should be located at complex interfaces, binding interfaces. That was the hypothesis. So we tried that. We applied this idea to the yeast proteome, and we generated a set of about 6,000 putative markers for complex binding interfaces. And then to assess whether these make sense at all, we performed an enrichment analysis, but this is not the classical geo-enrichment analysis. This is a domain enrichment analysis using exactly the sequences of the peptides that change. So not the whole protein, but the sequence of these markers. And these markers were enriched for protein binding interfaces. So based on the enrichment analysis, this is promising. The second term that popped up was nucleic acid binding. We did not expect this one, but we should have expected it, because in our pipeline, we probably separate also proteins from their complexes with nucleic acids. Now the data look like the example you see here. This is a pentameric protein. in ochre, you see the coverage, so the peptides we detect. This is a particularly well-covered protein. And in green, you see the peptides that change across fractions. And in light blue, you see where the interfaces are between the subunits. So this is a cherry-picked nice case, as usual. But then the question is, if you look more globally, is this any statistically significant? So to assess whether we capture interfaces, we could, in principle, start introducing mutations at the interfaces, because if you put mutations at the protein complex interface, you have a chance to destabilize it and disrupt it and separate the different subunits. That's an old principle. Luckily, we did not have to do this experiment because there is a library of known complex disrupting mutations. This is based on a manually curated analysis of many biochemical experiments in yeast. And 117 of these mutations were captured by our analysis, and 82% of these were exactly within our interface markers, suggesting that at least we are on a good track. Many of these markers are most likely at the interfaces of protein complexes. So what can you do with this? If you have an atlas of binding interfaces, you can use it for docking studies. So if you don't know how to dock to proteins, knowing where the interface is could be useful. You can use it to study the effect of disease-associated mutations. You can speculate on whether or not they affect a complex. You can also use it to decide where to introduce a mutation to potentially disrupt the complex you're interested in. And it can be also relevant for drug development. But what we are most interested in is the use of these markers for our classical functional proteomics screens. So whether now that we have these markers through a very tedious ultrafiltration approach, can we use these markers in a global analysis of an unfractionated sample to tell which complexes change their assembly state? Without doing any pull down, without doing the filtration anymore, can we just probe it directly in a whole cell extract? And now I will show you the last piece of data. So we tested this using cells treated with a hydroxyurea stress. This is a perturbation that triggers the formation of these inclusions you see here. These are called P-bodies, and they are phase-separated compartments. You can think of them as a big complex held together by low-affinity interactions. That's what a P-body is. And so we analyze these cells by a single LipMS analysis. So here, no fractionation, no filtration, just a LipMS of the cell extract. But then we intersected LipMS peptides with the library of markers for complex interfaces, basically to ask which of these changes is due to a complex assembly or disassembly event. And we obtained a list of about 50 complexes. You see some of the names here, whose assembly state is most likely altered under these conditions. And what is known about P-bodies, the formation of P-bodies, is that when P-bodies are formed, these two proteins here have to interact. So PAT1 binds to DHH1, which in turn binds to RNA. And this binding event triggers the coalescence of a P-body. And then other complexes are located into the P-bodies, the LSM complex and this other protein that binds RNA. So this is some of the complex assembly events that are known to occur during P-body formation. Now, what you see here is the sequences of all these proteins in gray. In yellow, you see the known interface based on structural studies. So this is all very well characterized by high-resolution structures. And below, you see the coverage of the lip analysis. So in gray, you see the peptides we detect. You see some proteins are not so well covered, others are well covered, others are extensively covered. And now in red, you see which of the lip peptides changes upon hydroxyurea stress and at the same time is part of the interface library. And as you can see, most of these markers are exactly at the known binding interface that is known to be regulated under these conditions. Here, here, down here. This case is very complex because, as you see, we don't cover all the subunits of this complex. We capture 1, 2, 3, 4 subunits. In most cases, the markers are at the known interface, like here, 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 and also here. But we also capture an additional change, which could be due to a new protein binding event somewhere, or it could be a false positive, of course. So this is what we are doing right now. This is work in progress, but I wanted to give you an idea of where we are going. And we believe that these markers could be very useful, if further developed, for the unbiased analysis of complex assembly and disassembly events directly from whole cell extracts. I should also point out the caveats of this analysis. You probably realize that LEAP depends a lot on sequence coverage, because whether or not you capture a certain change depends on whether or not you detect the peptide associated to that region. So important for the future will be to further expand the coverage of this analysis to capture also low abundance proteins. That's our classical nightmare in proteomics. We unfortunately have to lyse cells, so we hope that one day we will be able to do the digestion step in the cell. And LEAP is only one of the techniques you could use for such structural screens. There are also cross-linking mass spectrometry and different types of surface labeling techniques. I'm personally very interested in these two, and I think they are getting there in terms of global analysis. So it will be very, very interesting one day to couple these techniques to increase the resolution of this analysis and also the information content. And with this, I'm finished, and I would like to thank lab members who generated this data. This is work done by Chris Dörig and Kathy Marulli. They worked on the complex assembly project. Valentina Cappelletti led the osmotic stress project together with Ilaria Piazza and Thomas Hauser. Aleš Holfeld and Dina Schuster worked on the protein-protein interaction project. Lili Malinovska worked on alpha-synuclein, building on work done many years ago by Johan Feng, who left the lab. These are our collaborators and our funding sources. And thank you very much for your attention.
Thank you very much, Professor Picotti, for a very interesting talk. Now we're ready to take some questions from the audience.
Thanks for the talking. It's so amazing. And I have many questions. The first question is about the ProteaseK digestion time. I remember you have a paper on Nature protocol. In the protocol, the digestion time is 1 minute. So is there any difference for the 5-minute digestion time and the 1 minute? And the second one is for the molecule binding binding part. Do you have any clues for the size of a molecule? What kind of how big of this molecule can be detected? And the third one is, do you have any suggestion to optimize this to detect the binding the molecule binding
site
enzyme?
The binding site, you mean?
Yeah.
Okay, so I hope I remember the 3 questions. The first one was about the incubation time, right? You asked in the protocol, you suggest 1 minute. Now on the slides, I saw 5 minutes. I think that's what you wondered, what's best. So the fact is, it really depends on the organism you are studying. We typically try to optimize for a given species the number of lip sites we detect in a proteome. So that's the target, to increase the coverage again in detecting alterations. So that means that when you apply LEAP, you have to count the number of LEAP sites you can detect. And this we do by counting the number of half-tryptic peptides that appear upon addition of proteinase K relative to a classical tryptic digest, or the number of fully tryptic peptides that disappear upon addition of proteinase K. And that's how we estimate the number of cleavage sites. So for your organism of interest, you probably have to do a little titration of the protease amount and the time series to identify the sweet spot. So depending on the experiment and also the concentration of the lysates we are using, this can vary. But funny enough, we always end up hitting the same time range for many different species. So it's always between 0 and 10 minutes. So that's where we always end up. I hope this answered your question. Then the second question was about the size of the small molecules we tested. So I think the smallest things we analyzed were amino acids. So we added things like glycine, proline. This is the smallest things we have added. Now we are trying to ask if we can also detect metal binding sites. So we add metal salts, which is even smaller. And we are asking if we can detect the binding site. So in some cases, it seems to be the case, in others not. So I'm not sure what the answer is yet. And then the third question was if there is any way to optimize. Okay, see, yes, the binding experiment to detect the binding site, right? This is what you asked.
Yeah.
I guess you're asking because in my example, I mentioned that not always you detect only peptides at the binding site. Sometimes you detect peptides that are somewhere else. So the short answer would be no. There's no way to tell which one is the binding site and which peptide is completely unrelated and is involved in a conformational change. We had some initial observations that when you plot the affinity, you derive from the different peptides. Based on very few cases, it seemed like the peptides that were closer to the binding site had a higher affinity derived from this method. So maybe this is a promising approach, but we haven't tested it very, very thoroughly yet. This is just one of the ideas we have.
Hi, thank you for the talk, very interesting. I wonder about the data analysis out of these barcodes that you detect. Can you comment a little bit about the data analysis if you develop some workflow or informatics or something to analyze the data? And another thing is like I have the feeling that this method had some variability. So let's use enzyme 5 minutes or how. Yeah, I feel that the standardization could be a little bit tricky between labs. Let's say. Any comment about that?
Yeah, so maybe I start with the second question, not to forget it. So you are saying that you fear that since we are using unspecific proteases, I guess, you fear that there is higher variability, correct? In the data. Yeah. So that's something we wondered about as well. And also this was the main criticism when we submitted the very first paper. And so we had to repeat the same experiment 10 times with different operators to show also the level of variability across different people. And bottom line in that experiment, the message was the coefficients of variation are in the range of 15–20%, so very similar to what you see also in a classical experiment. And this was label-free quantification, so the CV is also affected by the fact that you are not using labels, right? But now what we see is that there is indeed variability when you change the batch of the protease. So if you buy again proteinase K, unfortunately, the level of activity is not always the same. So what we are doing now is to spike into every sample a set of peptides for which we know that the digestion ranges from very high to very low. Some peptides are completely digested by PK, some are not digested at all, and then you have all intermediate behaviors. And then we standardize, so exactly what you said, the activity of proteinase K in a given experiment based on this mixture. The other question was, what is the pipeline we are using? So, well, the database search is very standard. We only allow for the detection of half-tryptic peptides. Then we do quantification both by label-free quantification based on DDA or label-free quantification based on DIA. With DIA, we have to build first a spectral library that covers all the half-tryptic peptides that might be generated in your experiment. So you need to make sure that you analyze also the samples that have been treated by LIP. And if you have multiple conditions, you need to use them all for the creation of the spectral library. Now, what happens next is the big challenge, because for a long time we had to do the correction for protein abundance changes somehow manually in Excel, because in these samples you have at the same time changes in protein abundances and the changes in the LIP pattern. So if you have a proteolytic pattern, it could be that it's all shifted upwards or downwards just because the concentration of the corresponding protein changes. So we have been working with Olga Vitek now at developing an automated pipeline for doing this correction automatically and on a series of R scripts to then produce the barcodes automatically so that you already see the fingerprint for a protein and also to map the data to protein structures. This is the basically updated version of the protocol paper someone else mentioned before. This has been now accepted, so it should come out soon, the updated version of this.
Thank you.
Hello, very nice talk, thank you. You showed this experiment with 4 filters with different cutoffs, but did you also consider actually doing like higher resolution fractionation, like for example, using size exclusion chromatography, because you might lose some complexes which are, I don't know, like 250 kilodaltons and then change to 150, or that would be just too much work?
No, you're absolutely right. We considered actually using size exclusion chromatography. One big concern was the dilution effect you have by SEC, because the fractions you obtain are very diluted. So we feared that several complexes or partly assembled complexes would be a bit affected by this dilution factor. But you are right, this would be another way to do it. We haven't done that.
Thank you.